Good afternoon. This is Dula Amarasiriwadana. I'm professor of chemistry. I am moderating this second session of environment and change. And so we had a wonderful session in the early afternoon, and we have 6 speakers ahead. And without going any further, we will start with Aidan McGregor. And he's going to talk about The Bow and Arrow, a novel, which will be talking about the outdoors, okay? So Aidan, and I will be also give a warning sign, you know, it's a 10 minutes talk and 5 minutes question and answer. And about 8 minutes also, I will give you a warning. I'll raise my hand, okay? 1, 2, like that, okay? Thank you. And Aidan, it's all yours.
Thank you, Dula. Hi there.
Hi.
Hi, everybody. My name's Aidan R. McGregor, go by he, him pronouns. And for my div project, I wrote a novel. I wrote a creative piece that I've been thinking about and sort of writing for the last 10 years. Though 85% of it was done in the last year or so, thanks to my wonderful advisors, Jeffrey Whalen and Alex Cuellar. They're just a couple of gentlemen who really helped me a lot, and that's not a word I use very often, gentlemen. I also want to thank my parents for their unceasing support of my creative aspirations, and my sister for constantly helping me exercise my imagination as kids when we were playing with little elven figurines and telling stories that neither of us remember. So this novel, I really love high fantasy. I always have. I have read Tolkien's The Silmarillion at least twice, and I love it. I'm that kind of dork. And but the kind of writing that really impacted me as a kid was naturally young adult fiction. Writers like Rick Riordan with the Percy Jackson series, Heroes of Olympus, John Flanagan with the Ranger's Apprentice series, and Aragon, The Inheritance Cycle with Christopher Paolini. They were all stories that I grew up on. And so while I one day hope to create a mythos, a basis of stories as big as Middle Earth's, I don't want to present them in a Silmarillion or a Lord of the Rings, which is great because I don't have the time or skill, especially in this last year, to write a book like that. I want to write shorter young adult fiction stories that follow characters through struggles of their own. So I thought I would start off by reading a synopsis of the book that I wrote. And right, so here goes. I don't have any presentation for this, I'm sorry. So Victoria, an English teen who has mastered such solitary tasks as bow hunting, camping, and adventuring, but also has a penchant for watching Netflix and procrastinating on homework, is suddenly forced to survive alongside 8 people that she has never met before. When they all find themselves abandoned in a magical forest, seemingly universes away from Earth, they look to her and her knowledge of survival skills, and they prove more important than any magic. Surviving is difficult with so many mouths to feed, but it is made even more difficult by various creatures and forces that seem to be hunting them. Territorial wood sprites, diaphanous spirits that share their faces, and a huge, terrifying tree monster with rotten vines and poisonous berries. As they piece together why they are there, they discover artifacts key to each of them and granting them elemental powers, as long as they pass the difficult, dreamlike trials that pry at their deepest perceived shortcomings. And next, I'm going to read a little excerpt from it, a short section from the beginning of the book. Like I said, I don't have a presentation for this, but if any of you would be interested, I'm posting a link that should be accessible to all of you in the chat. That would take you to exactly what I'm about to read. But I'm sorry, if you can't read it, if you can't access it for whatever reason, my apologies. I just threw this together last second. And but if you ever do want to read this, it is up on the Hampshire College archives. So as long as you remember your Hampshire emails, you can access it there, I think. So, The Bow and Arrow, the novel. Bluebells. In the idless woods, the ground was saturated with bluebells that weaved in between the blades of grass. They were so lively and vibrant that she thought they might be growing between her toes, even as she stood there. Victoria took a deep breath, her hands shaking. The target was out of place in the sea of blue. The outermost circle was white, then going in, black, blue, red, and yellow. Victoria's left arm was throbbing from the tension, but she would not allow her fingers to slip. The ground around the target was covered with arrows, like tiny saplings. At last, the tip of Victoria's arrow was exactly where she wanted it. She released the tension on her hand, and the arrow was away. Just as she fired, a gust of wind rose up, and the bluebells seemed to turn and watch as Victoria's arrow was taken away by it. She let out a frustrated, "Ch! I really thought I had that," she grumbled. "Me too," said her father. Eli was sitting on a tree stump behind her and observing her form. "Your release was better this time, so good job. What went wrong?" "The wind," she said defensively, gesturing with her bow. Then she lowered it gingerly, remembering that it was her birthday gift. Eli frowned, then said lightly, "I didn't feel anything." He extended his long arms exaggeratedly like he was a sail on a log-sized boat. "Well, you wouldn't have. It was over here, wasn't it?" Victoria shouted in frustration. It was only afterwards that she heard her own voice. Oops. Eli's frown deepened and his tone became a little lower. "Should we be done for the day?" No, Victoria backpedaled. "I'm good, we're good." "Good," said Eli evenly. Victoria glared at the target. "I just thought I would at least hit it." Eli chuckled and his tone returned to normal, though it was heavy with sympathy. "It's normal for the first try, if that helps." Victoria lowered her eyes to the bluebells. "It doesn't, really." Eli sighed, but then he said, "I missed for weeks when I was starting out. Does that help?" Victoria slowly smiled. She looked back at him and said, "Yes, actually, it does." Eli stood up from his perch on the log and stretched. Yawning, he said, "Provide positive encouragement?" "Check. I'm hungry. Do you want anything?" They were at Eli's log cabin in the woods. Late spring sunlight tumbled off of flower-laden branches and landed on their thatched roof. Bluebells grew all the way up to the uncut flagstone below the small door. Moss grew on the rocks on the south wall, and at least 2 generations of finches had grown up between the cracks. The cabin was just one room, practically a storage shed really, but it felt like a home. It even had a hand-carved sign which read, "The Morgans." The only thing that stole from the quaint, rustic beauty of it all was the small solar farm around the corner and the satellite dish on top. Eli began walking up the path to the front door and looked back, repeating himself. "Hey, Vic, are you hungry?" Victoria felt her stomach grumble, but she looked down at her bow. "I don't have to stop, do I?" Eli smiled. "Now, why would I make you stop? You almost have it." Victoria beamed as Eli turned and went into the cabin. Victoria reached down, expecting to find an arrow sticking out of their makeshift quiver, but then she realized she'd already fired them all. She looked around the clearing at the many missed shots and groaned. Eli had a rule that you have to pick up all the arrows after a set, so she couldn't just grab a couple and keep shooting. She'd have to gather them all, and if she didn't, he would know. Her arm was throbbing with muscle ache and her fingertips felt hot. Was going to pick up all the arrows really worth it? She couldn't do this for much longer anyway. Victoria shook her head. Today was a special day. Today was the day she turned 10 years old. Today was the day she had gotten her own bow, and the day history would remember as the first time that she, Victoria Morgan, had ever fired it. And if she wasn't too careful, she would remember that today was her first birthday without her mother. Naturally, that wasn't something she aimed to remember. Her fingertips twitched, her eyes narrowed, her sore muscles ached for more. "I'm not going to stop now, not when I haven't even hit it." Victoria swept through the clearing like a second wind embodied, picking up every last arrow and putting them back in the quiver, which was really just a PVC pipe glued to a half log, and the purpose of such an apparatus isn't immediately clear when it's empty. For instance, the first time she'd ever seen it, Eli picked it up by the pipe end and told her it was a hammer. Victoria selected the arrow which she had subconsciously picked as her favorite, a red one with orange, yellow, and white fletching. When it flew and spun, it looked like it was on fire. She straddled the firing line, which Eli redrew every day instead of fashioning a more permanent solution. Then she took a deep breath. The afternoon sun cast light on the ground, making a colorful pattern on the bluebells, and as the trees shifted in the wind, the bluebells seemed like an ocean under the clouds. Victoria knocked the arrow to the bow. She felt the wind part over her arms and her arrow, and felt the grass and the bluebells tickling her bare feet. Victoria drew back the drawstring and breathed out at the same time. Sunlight reflected off the shaft of the aluminum arrow, sending a ray of red light into the forest. The wind died, and a hush fell over the forest, as if awaiting her next shot. Victoria released, and the arrow whistled as it flew through the air and smacked against the target with a distinctive slap. Bullseye. Victoria stood there stunned. Eli ran out of the cottage, still holding a jar of Branson pickle and a butter knife. "Hey, that sounded pretty good. Did you—" He trailed off as he looked back and forth between Victoria and the target. Dropping the jar, Eli grabbed Victoria and flung her up in the air, shouting and ululating. Victoria giggled and whooped as well, and they danced together on the bluebells. Thanks for reading along. Thanks for listening. that's my presentation. That's my project.
On time. Thank you, Aiden. Now it's open for questions, and please do send questions, chat, or anything like that. While we are waiting, what's the basis behind this story? How it's from your personal experience, or are you a very good outdoorsman? No, something like that. You know, there must be some reason for that. Yeah.
I mean, I love archery. Some of those experiences
I see.
are things that I have, but I really don't share much with the character in terms of survivability outdoors.
I see. How long it took to make this story?
The characters first started. I first started making up the characters 10 years ago.
10 years ago. Oh, I see.
I've begun this. The stuff that I just read is probably some of the oldest writing in the project that still is in the project, because
Right.
I wrote it and felt happy enough about it to keep it, but the rest of the story has gone through versions that have never extended beyond like 25% of the whole plot.
I see, I see.
So
you're a good storyteller.
thank you.
Yeah. Any questions to Aiden? And so what are you going to do next?
There's a question in the chat.
Yes.
Yeah.
That'd be my parents. So like I said,
Can you tell us about the designing and fantasy map and writing an imaginary language? Yes.
That's an aspect that didn't make it so much into this project. The fact that I've worked on making up fantasy languages for the world that Victoria and the others end up in. And I really enjoy drawing maps, and these didn't end up getting into the book either. But that's just something that I love doing. And I always tell people who are trying to recommend books to me, I say, you can be sure that I'll like the book if it has a made up map at the beginning and a glossary of made up words at the end.
I see, I see. So what are you going to do after this, once you graduated?
Well,
are you going to write novels and publish or something like that?
Absolutely. This is the first story in a series that I've imagined writing for a long time. That's always been the plan when we were discussing in meetings with my two advisors, gentlemen, as I said earlier, Alex. Always making it clear that this story sets up some of the primary characters for a bunch of stories that I want to tell.
Right. Fabulous job, huh? Thank you.
Thank you all.
Yeah, I do get a lot of accolades here. You can read that.
Oh yeah, question.
Yeah, there's a question. Your use of language is so beautiful and descriptive. Yeah.
Oh, I suppose I've already answered the question a little bit. Yeah, I imagine this is a series for sure.
Yeah.
But this book isn't. I finished a draft, to be clear. I finished a draft this year. And my mom wants me to bring the map closer.
Oh, that's right. Yeah. Okay. That's that. What is the map of that one?
Oh, just a made up place.
Okay. Wonderful work, cartographer.
Thank you all. And sorry for the confusion.
Thank you very much, Aiden. Yeah.
Been in this showcase, but I'm really glad I still got to present. Thank you all for listening so much.
Take care. All right. And the next speaker is Rose Madison. Rose is going to talk about white bark pine management in Glacier National Park. Rose.
Hello. All right, so I'm going to go ahead and
Oh, yeah. Yeah.
All right.
Yeah, and open up completely. View mode. Yeah.
All right.
Yeah, great.
So I remember a few summers ago talking to a family of friends who had recently gone on a hike in Glacier Park, which is very close to where I lived. And she said that this crazy thing happened when she had posted on Facebook about it, that she hadn't posted a picture or anything, but she just mentioned this tree on this trail, and she got so many people responding to her, talking about how much they loved that tree, sending pictures of themselves hugging that tree. And she was like, how did they, like, they knew exactly which tree I was talking about. And the funny thing was, I was like, oh yeah, I know exactly which one you mean. And it was actually this exact tree. And I really like thinking about that memory, especially throughout the project, because it's one of many that has shown me as someone who lived in the area, and also just reminds me how important this tree is, not only to the ecosystem, but to people as well. Yeah, so like what Dula said, part of my div was on management of whitebark pine in Glacier National Park. So this is a map that I made. The lined parts show the range of whitebark pine. Whitebark pine grows in subalpine and alpine areas of the Western US. And that small little blue part that you can see is Glacier National Park, and the part next to it that's colored is the Blackfeet Indian Reservation. In my full div, which included a project proposal, a management plan, and a creative writing piece, I did a fair amount of work about talking about how whitebark pine is important to Blackfeet people, and actually most of Glacier Park used to be their land. So that is kind of a big part of this, and I'll come back to that later in the presentation. So first of all, what is whitebark pine? So like I said, it grows in subalpine and alpine ecosystems throughout the Western US and Canada. It's a keystone species, so it basically supports the ecosystem in more ways than you would expect. Some examples of this are through supporting watershed health, through soil stability, and many animal relationships, including grizzly bears. You can actually use years that have high harvest of whitebark pine seeds to predict if there's going to be a lot of interaction between humans and grizzlies, because if there's a lot of seeds, they'll spend more time in these really remote areas eating instead of having these encounters with people that can usually be harmful for both. One of these most important animal relationships that whitebark pine has is the Clark's nutcracker. This is the one bird that has evolved to crack open their hard cones. And what they'll do is they'll cache a bunch of seeds in one spot, usually behind either a log or a rock. And what this does is it allows whitebark pine to help initiate what's called tree islands. So tree islands are basically area above the continuous timber line. They're patches of vegetation, trees, bushes. And in Eastern Glacier Park, there's a lot, whitebark pine is the main initiator of these tree islands. So what will happen is, because of being planted behind a rock or a log, some kind of nurse object to kind of protect it from the elements, it'll help the tree grow, and that tree growing will create favorable growing conditions downwind from it, which will allow more plants to grow and will make this positive feedback loop of good growing conditions till you have these little islands. But one issue that we've been seeing is whitebark pine has been dying back at a tremendous rate. And one of the biggest reasons for this is white pine blister rust. So blister rust is a fungi-caused disease that's spread through wind. And what will happen is it will attach to a limb, and it'll start to cut off nutrients on that limb and slowly work its way up to the trunk until it kills the whole tree. And a few signs of this include blisters, such as in this picture, and flagging, which is basically just red needles, which you can kind of see in the background of this picture. And it's also ribes species, so gooseberries, currants, things in that family are secondary hosts, because blister rust needs multiple hosts in its life cycle. So you'll see higher infection rates in areas that have higher humidity and higher rabies species. And another issue that plays in with the blister rust is mountain pine beetle. Mountain pine beetle and whitebark pine have lived together for centuries. So generally when we see outbreaks of mountain pine beetle, it shows dire conditions such as hot, warmer conditions in the 1930s. Now with climate change, which I'll talk on more later, and you'll see in trees that have had pine beetle, you'll see these galleries, these kind of markings on the tree, like the one in this picture. And it's not completely known yet if the relationship between blister rust and pine beetle, but it's thought that if one is attacked by the other, it's more susceptible, but that needs more research. Another big thing that's been leading to dieback of whitebark pine is fire regimes. So we've been doing widespread fire suppression in the US since the early 1900s, and that's really harmed whitebark pine because it really relies on these fire regimes. And because it's not a shade tolerant tree, so it can easily be outcompeted when there's not fire, but when there is fire, because of the Clark's nutcracker spreading the seeds, it tends to regenerate really well. One area that might be interesting to kind of learn more about regeneration effects is the St. Mary area of Glacier Park, which is pictured during the Reynolds Fire of 2015, because there is some regeneration happening in this area. And probably the biggest thing of concern, on top of everything else that's causing this dieback, is climate change. Climate change is very much impacting everywhere in the world, but especially Glacier National Park. And one of the easiest ways to see this is through the glaciers that are shrinking. So this picture is of Sperry Glacier, and it just tracks the melting rate over the years. And this warming is also really impacting whitebark pine. And what it'll do is whitebark tree islands are really important with climate change because the vegetation is supposed to move up in elevation, which is what's predicted. But it can't do this without being outcompeted from other species moving up if we don't have these tree islands to help do this. And so that's one of the reasons why whitebark pine is even more important now than it ever has been. So there's a few different things you can do for restoration. None of them have really been the perfect solution, but one of the best is planting programs. The first thing is finding blister rust resistant trees and collecting seeds to set up breeding programs. This is a picture of putting netting over a cone just so that they can collect it without any other creatures eating it. There are some areas that have set up pretty good breeding program protocols, such as the Coeur d'Alene Forest Service, but there is more need in this area, more work that needs to be done. Another common suggestion is using prescribed burning to help restore whitebark pine, but this can be expensive. It's hard in remote areas where whitebark pine grows, and it can also easily get out of hand with the fire suppression we've had. So my recommendation instead is when wildfires burn in these areas, let them burn, but of course, protect people and buildings as needed, but let them run their course. And the biggest thing, like with any issue, is education and policy. So there needs to be a lot more education for the public and ways to get involved with whitebark pine. That was actually one part of my div, was a creative writing piece that is meant for the public and tells ways that individuals can get involved. We need more media, such as this book I found when making this presentation, to teach people of all ages. We need to work with local people on restoration projects and also local Native peoples, and not just take their knowledge, but actually respect them and their knowledge in the lands and actually make sure they are truly a part of the project. Another thing is So next steps, there's a lot of research that still needs to be done. There's a lot of restoration that needs to happen, and we don't know a whole lot about these effects. So one thing that I was going to do originally as my dip was I was going to research the impacts of blister rust infections between tree islands and continuous timberline but that didn't happen because of COVID closures. So I hope to do that in the future at some point. And there just needs to be a lot more research on ecological interactions with all these causes of dieback and white bark pine itself. Like I said earlier, things such as interactions of blister rust and mountain pine beetle. And that's it.
Thank you. Thank you, Emma. And it's now open for questions. You can fire questions on chat line and Q&A.
Oh, okay.
Thanks for reading us. I have a question for you, actually, too. You know, last time I went to Glacier National Park, I think it's most of the things are melting away, you know, many glaciers. How long it takes to establish this white pine?
For white bark pine to establish?
Yeah, establish,
So,
yeah. Days, years, decades.
It is a slow growing and long living species. So it doesn't reach reproductive age till about 30, but it often takes closer to 100 years till it actually has enough crown space to create a good amount of pine cones. So it does take a long time, and that's part of the issue, is it's not just something that you can grow and quickly have it producing cones and keep growing the population. So that's part of why it's something that needs to be worked on right away. It's actually, blister rust has been studied for a long time in other white pine species, but those were ones that were more important for lumber industries. So white bark pine kind of got it a little later because it isn't valuable wood and it is in more remote areas.
I see. Another question is that you said there are a lot of fires, right? And they firefighting chemicals, right? So I know most of the chemicals are ammonium phosphates type, you know, most firefighting chemicals that they threw that you showed in the picture. Do you see any eutrophication on those lakes? I know there are a bunch of lakes up there. Yeah.
I don't know how the fighting efforts have affected that. That's actually a very good question. Yeah, I don't really have a good answer to that.
That's okay, yeah, yeah. There's a question, yeah. Do you have a website where we can read your creative writing?
I don't have a website. I think I submitted it to the archives. I actually don't completely remember because I graduated in December. But if I have it, I'll check later today and make sure that is in the archives for people.
Yeah, yeah. You can access through the library, right? Yeah. Yeah. Any other questions? Everybody say thank you, very good. So what are you going to do, Rose, after this?
So I spent a very long time looking for jobs, and because of COVID, having very little success for a while.
Yes.
But in a few weeks, I'm going to start work at a biology lab, actually, at Stanford, doing work with non-coding genes and thyroid cancer. So not exactly my field, but it's techniques that could be really helpful with things like blister rust. And in general, I think will just be really interesting and important work. So that's my plan for the next couple of years. And then after that, I plan on going to grad school to hopefully get a PhD in ecology or a related area.
Wonderful. Now, there's a question. Rose, I know you spoke about the research you would like to do in the future. What do you imagine as a future project, collaboration with others?
I did,
We lost it.
Oops.
Yeah, we lost Rose.
We lost, yeah.
I'm here for tech support, but there's not much I can do about losing
All
someone.
right, okay. I think we can go ahead. Yeah, thank you, Rose. Yeah. We are ready to go the next one. I think we lost Rose, but thank you very much.
Yes, so we can move on to Hayden.
Yes, I think we are running out of time also. The next speaker is Hayden Gadd, and he's going to talk about ALS disease pathology through the microglia. Thank you. All yours.
Thank you, Dula. First, don't mind the fact that I'm dressed like a pilot right now, or that I'm on Bear Mountain behind me.
That's great. Yeah, that's great. Yeah,
I'm getting senior pictures later, so I didn't dress for the occasion. Not that it's not important to me, but let me share my screen really quickly. Can you all see this? See this and all looking good? Okay, great.
Yep, it's great.
Okay, so my name is Hayden Gadd. You see him pronouns. And this is my div, focusing on a certain kind of brain cell and its relation to ALS. My div 3 journey has been a little bit idiosyncratic, like I feel like pretty much everyone else's has. Just got a call from an 800 number, that's great. But I actually started my div 3 back in the last spring, before it got cut off by something. I'm blanking on what it was. And then I decided to opt out of last semester because of concerns about that thing that's escaping me, before coming back for this semester to finish things out. So a lot of this kind of came together this semester. I'm gonna walk you guys through exactly what I did. So before we even talk about ALS, it's probably a good idea to, let me get a laser pointer here. Probably a good idea to walk through exactly what ALS even is. So ALS stands for amyotrophic lateral sclerosis. What does that mean? So to break that down even further, amyotrophic means atrophy, atrophic, of the muscles, myo. So muscle atrophy caused by lateral scleroses, which scleroses are scar tissue, and lateral just means the lateral parts of the spinal cord. So breaking it all the way down, it's atrophy of muscles from scleroses forming in the lateral parts of the spinal cord. But we'll keep it to ALS. Some symptoms of ALS include a gradual loss of voluntary motor control, so not being able to move your limbs, your neck. Eventually, some people have affected cognitive or behavioral function. Many people say it's harder to think or have odd behavioral changes that they're not conscious of. Notably, it only affects motor neurons, so neurons that are part of your sensation, your taste, your touch, your sight and smell, not affected generally. And it affects motor neurons with a few crucial exceptions, including most notably eye movement. Your eyes can still move, and that's because the nerve controlling your eye movement is actually farther up in your brain, whereas ALS starts to form either in your spinal cord or in the back of your brain. And unfortunately, it is a terminal illness. Death typically occurs within a few years, with some exceptions, most notably Stephen Hawking, who lived for about 50 years, I think, after his diagnosis, and also guitarist Jason Becker, who I believe was diagnosed in 1989, and to this day, still, through a system of eye movements that his family is able to translate, he still composes music, which is rather incredible. I would really check him out. So to get into the nitty gritty of all of this, I'm not gonna lie to you, genetics and neurodegenerative diseases are really complicated, and it gets really messy really quickly. Because if it were easy, then we would have found cures by now, we would have treatments. So like any other neurodegenerative disorder, or most neurodegenerative disorders, we don't really know a direct cause of ALS. There's not like an ALS gene, that's the single one that causes ALS, or even like a single system that causes ALS. In fact, many of the genes that we associate with ALS, if we take a mouse model of ALS and take those genes out that we associate with ALS, the mice typically die or have some sort of disrupted cognitive function. So we know that all these genes are crucial for our life functions. And really the hallmark of ALS, as well as many other neurodegenerative disorders, is this clumping up of proteins inside of the cells, this protein aggregation. For multiple possible reasons. These aggregations are called inclusion bodies, and they're really the main reason why cells start to function differently, or they end up just dying, or immune cells end up killing them. And to give you a visual of what this looks like, I just pulled this from a paper. This first column, we have cell nuclei. Second column, we have proteins tagged with a green fluorophore. And then the third column, or the third row, my apologies, are the combined images. So this first column, we see that there's not really any big aggregations. There's little protein pinpricks here and there. This second column, however, you can see these large clumps of proteins that are affecting all of these cells. And those are what inclusion bodies look like with fluorophores under a microscope. So getting into my research a little bit, I looked at two specific genes that are related to ALS. TARDBP, which encodes for the TDP43 protein, and UBQLN2, which encodes for the ubiquitous protein. Now, I'm not going to quiz you on any of this. You don't have to remember what these things are. These are just the two things that I looked at. So TDP43 is important in ALS because 97% of ALS cases, the protein that gets clumped up is TDP43. TDP43's role is actually an RNA function inside of the cell nucleus, but that doesn't really matter for ALS. The big thing is that it's the protein that gets clumped up. Ubiquitous is actually involved in protein degradation, which you might think, well, how is that involved in ALS? The hypothesis is that if there's too much ubiquitous, it latches onto the proteins to tell the machinery to break them down, like, hey, come here. But if there's too much ubiquitous, then it won't let go, and the protein can't, the degradation proteins can't attach to break them down. So you get these big clumps of TDP43. So interesting about these two genes is that they're ubiquitous. They're in every single cell in your body. They're not just in your neurons. They're not just in any specific cell. They're in every single cell in your body. In this paper that I read from 2019 showed that inside of neurons, if they overexpressed ubiquitous, that is, they made the cells have a lot more of this protein, these TDP43 inclusion bodies formed. So after I read that, I thought, well, why not other cells? Do they affect cells that aren't neurons? So in comes the microglia. So the microglia is the resident immune cell of the brain. It's involved in mostly everyday upkeep. So cellular debris, it eats that up. If certain parts of a cell aren't being very effective at their job, they'll prune those away. But crucial in neuropathy research is its role in first-line disease fighting. And that's because in your body, you have immune cells that are very specialized. They have very specific roles for very specific things. But those cells have a hard time reaching your brain for a couple of reasons. So microglia are the first ones. If there's a bacterium or a virus, or if there's a cell that's misbehaving, then they're the ones who typically they both signal your body's immune system, and they also they'll also start fighting the cells themselves. However, in doing that, they tend to kill all the cells around the cells that they are targeting, because they just release cytotoxic factors just, willy-nilly, essentially. And they're not very specific to specific cells. So they end up killing a lot of cells, which is why a lot of neurodegenerative diseases are as impactful as they are, because of these, you have a lot of cell death as a result of this. Now, other research has shown that in ALS, these gene mutations that are associated with these aggregate formations are also mutated in the microglia. And in other lab studies, they've shown that these microglia they misbehave when these genes are mutated. They end up releasing more factors that could kill other cells. And now the unfortunate thing about microglia is that they're the least researched of all the brain cells. And that is mostly because we discovered them the latest. Because it's very possible that we haven't even discovered every kind of cell in our brain. But these ones, for a long time, it was believed that they came from the body and migrated to the brain. But only recently, within the past couple of decades, they realized that, oh, wait, they come from the brain. So not a ton of research has been done into them, which is rather unfortunate for, the main immunological cell of the brain. So anyways, I wanted to see if these inclusion bodies could form inside of microglia, because if these genes are ubiquitous throughout the body, and these ALS mutations can occur inside of microglia, then I wondered if inclusion bodies could be able to form in microglia and potentially add to the dysfunction associated with ALS. And I also wanted to see the connection between ubiquitous and TDP43 in the neurons. So picture time. I got some microglia, these BV2 microglia, and I split them into two separate lines. One of them I added a ubiquitous 2 plasmid 2, which essentially transfection just means I put the DNA inside of the cells, and then those cells would then produce more of that protein. And in the control line, I added what's called an empty plasmid, which is just, it doesn't do anything specifically to the cell. I just put it there so that it would also be transfected. I let those sit for a couple of days, and then I added antibodies to them. I fixed them with formaldehyde, and then I added on the experimental line this ubiquitous 2 antibody that glows green and this TDP antibody that glows red. And then in the control line, since I didn't add a ubiquitous 2 plasmid, I just had the TDP that glows red. And I did this a bunch of times, and I imaged and measured the results afterwards. And so what I saw from that, as you can see here on the left, I have the treated microglia, and on the right, the control microglia. And the green represents ubiquitous 2, the red represents TDP43, and the blue are just the nuclei of the cells. So if you can see in the treated group, we would really be looking for like those large clumps of TDP43 inside of these cells, which we don't really see here. You can see them inside the cell nuclei, but that's where they're supposed to be. As you remember, they are associated with RNA function. But you don't really see any huge clumps inside of these processes of the cells. So that was sort of step one. I realized there was no inclusion bodies inside of the cells. I did find, however, an association between ubiquitous 2 and TDP43. As the treated dish had more cells expressing TDP43, and all of those cells expressed TDP43 more. They, for measurement purposes, they glowed brighter. They expressed more of it. So what I drew from this, unfortunately, I found that there were no inclusion bodies within the microglia. Or I suppose fortunately, I'm not really sure how to look at that. But I did find a positive correlation between ubiquitous 2 and TDP43 production. Now, as unfortunate as it might sound that I had a negative result, it kind of just confirmed what the field already thinks, which is that ALS is rather complicated. A gene association that can be found inside of neurons that could form TDP43 inclusion bodies doesn't necessarily translate to microglia. I also ran these experiments on mouse fibroblasts, and I found similar results. There was a positive association between the two, but no inclusion bodies were found. So again, it sort of just already confirms what the field already believes in the first place. And so that essentially was the conclusion of my research. So thank you all for listening. I have a couple extra pictures of the microglia here. This is after I starved them for about 3 or 4 days, so they were rather unhappy. I'd also like to, that list of acknowledgements. I really appreciate everyone who helped me with this research in whatever possible way. That's it.
Thank you. It was a great presentation, yeah. So what are you going to do after this, Hayden? Mm-hmm. What are you going to do?
Oh, what am I going to do? Oh,
yes.
Yes, I'm intending on starting research in a neurology lab at UMass Medical. Technically, I haven't had an official job offer yet, but I'm already on their mailing list. Like the PI is intending for me to work in the lab. So that's what I'm planning on doing. I'm thinking about doing that for a few years, get more neurology wet lab experience, and specifically with microglia. This is a microglia lab. And after that, I'm intending to get my PhD in neuroscience. Probably something more specific than neuroscience, but hopefully something with glial cells, glial cell research, something of that sort.
Any questions? You can send those questions to Hayden. We have almost 3:45. Yeah. I have one question for you, and then we are all good. You know, you have these beautiful images, you know, and can you some sort of you count those images, right? Images.
Mhm.
If there's a way you can look at the intensity or something like that to more quantifiable way, people do that, because I'm not much familiar with this kind of research, but I just asking, yeah.
Yes, absolutely. There, I actually did do that, which was the second graph that I had on there. Essentially, you just you take the images, and I use a software called ImageJ, and you just circle the area where they're located, and then you quantify the pixel intensity from that.
Okay, I see. So you can kind of calibrate it to get some idea with known ones. Okay, that's cool. Yeah, thank you very much, Hayden. Yeah, all the best.
Oh wait, one sec. There's a question in the Q&A. Fiona, there wasn't really a big interest in ALS from any source, really. It's just that I'm really into neurology. I really like disease research. Well, I got interested in microglia, and as it turns out, microglia are heavily implicated in ALS and all other neurodegenerative diseases, like I said. But yeah, so I just kind of rolled with glial cells.
Any other questions? If not, thank you very much. It was great. Yeah. Yeah. And we are now going into the next speaker. Thank you, Hayden. And the next speaker is Kai Valieri. He's going to talk about new language. So we'll see what's that it is. íkjanísh, translating the climate crisis.
Thank you.
Kai is there?
Yep, I'm here. Hello, everyone.
Cool. Yeah, I'd like to hear your language.
Yeah, I will be speaking it. Probably.
Yeah.
All right, so my name is Kai. Let me share my screen. Let's see, I think this is the way. Okay, well, I've never shared my screen before, but looking good. All right, can everyone see the presentation?
Yeah. Can you make it bigger? Yeah.
Yes.
Loading, still loading.
All right, there we go.
Lovely.
All right, so my division project. Okay, so my Division 3 project is an ecolinguistic research paper that consists of my efforts to motivate, build, and test the claim that we need a new language to help treat the climate crisis. My approach centers around the development of my constructed language, íkjanísh. But before I go into detail, however, I would first like to thank my family and friends for showing up today. Your continued support means the world to me, and I'm very excited to be sharing my work with you and the broader audience today. I'd also like to extend my deepest gratitude and appreciation for my co-chairs, Ashley Smith and Blair McLaughlin. The amount of care, attention, and concentrated focus they brought to my project cannot be overstated. So for the project itself, the central argument, again, is that we need a new language to help treat the climate crisis. And I formulated and developed this argument based on the following premises: that language, culture, and thought are deeply intertwined, that grammatical structures and cultural influences work in tandem to habituate people to favor certain language patterns over others, that proposed responses to climate change frequently rely on language rooted in problematic frameworks like hierarchism, objectification, and restrictivism. More on that soon. That the sociocultural and historical context of natural languages significantly complicate their capacity to remedy this issue, and that a new language, here íkjanísh, can draw attention to how prevalent language forms encode particular conceptions of the world and potentially help shift them. I've come to the understanding that language is critical to how we respond to climate change by way of extensive scholarship on the intersections between linguistics, ecology, and environmental attitudes, an area of study referred to as ecolinguistics. So ecolinguistic literature points out that prevailing modes of discussing human-environment relations are often rooted in the same cultural frameworks that have enabled and driven key socio-ecological challenges like the climate crisis. So looking here at the stories we live by, as comes from ecolinguist Aaron Stibbe, who categorizes those types of problematic frameworks as stories we live by. According to him, the stories we live by are cognitive structures embedded in the minds of multiple individuals across a culture and influence how they perceive, speak, and act in the world. So they're kind of subconscious mental models that have profound ramifications in terms of physical policies in the world of how we interact with the environments. In this project, I focus on and classify 3 stories that are very common in Western culture and emerge in a lot of proposed responses to climate change. So I won't go through these all in depth, they are on the screen, but in this presentation, I'm mostly going to look at objectification and an example of how my language is meant to interact with it. And objectification is the pervasive tendency to consider particular entities as inanimate objects, which allows for them to be owned, used, controlled, bought, sold, and traded, and kind of paves the way for these large projects of extraction, ecological exploitation that have been contributing to like the climate crisis and biodiversity loss and habitat destruction, all sorts of things. So Aaron Stibbe says that the stories we live by are actualized through language and can be actively distinguished and shifted through conscientious and strategic engagement. And I contend that a new language can be uniquely beneficial in this regard. So as a new language or a constructed language, íkjanísh is, other examples of constructed languages are like J.R.R. Tolkien's Elvish languages in The Lord of the Rings, or the auxiliary language Esperanto, which is designed to act as a universal medium for like communication between languages. íkjanísh is kind of different from those in the sense that it is not always to be considered a language. I argue that íkjanísh is more of a language-based tool, and I've come up with the term utility language or Utililang to kind of encapsulate that meaning. As a Utililang, íkjanísh is made to be used as a means to evaluate existing discourses on the climate crisis and human-environment relations and to help formulate new ones. It becomes a tool to help shift our collective consciousness away from problematic models like hierarchism, objectification, restrictivism, and toward more sustainable imaginings. So some of the pieces of the language as I've constructed it so far, and it's not finished, so there's still a lot of work I want to do on it, but there's a lot of pieces I have put together that have allowed me to demonstrate it as a tool. On the left here is kind of the alphabet and the sound system, and that's just important to get a visual understanding of what it looks like. It's not something I use a lot in the project, so I thought that might overcomplicate things. And on the right, there's a list of some of the key words I've come up with in my language. And the word íkjanísh itself is translated literally as for imagining our shared homemaking. So even in the title, it's meant to be seen as a tool for a specific purpose, and that is to kind of reimagine how we exist in the world and what kind of choices we make and ways of interacting with each other and with our natural environments. And they're not so separate. That's a key principle of íkjanísh. And that is seen in the word wanish there, which is kind of the equivalent of environment or nature, but includes very conscientiously human and other than human beings and processes. And in íkjanísh, there's a particular part of speech I built into it to foreground the notion that everything is intrinsically interrelated. And these are a concept known as relations. So they're mandatory in íkjanísh sentences. They come after the main verb and they change how it's to be interpreted. It's built on the concept of grammatical gender, which is in linguistic terms is kind of a way of classifying different nouns. Like in Spanish, there's like masculine and feminine nouns, and in íkjanísh, it's animate and inanimate and conceptual. And animate is used for it by my purposes. It's all very subjective, but it's used for things or beings that are alive and able to act on the world in their own way, whereas inanimate objects are an inanimate grammatical gender categorizes something as functionally inert and not really able to do much without like interaction from an animate being, such as like a pencil could be an example. And so this is with the verb to plant, which will come into play soon. And so I'm not going to go through all of these, but the first one, which is pronounced ok, is between two animate beings. So the event of planting becomes a way of facilitating growth. So these two beings are in relation. Whereas the one oldek, where the thing being planted is inanimate, it's more of an investment that the animated being is is initiating. So as a tool, this is kind of how I conceive for íkjanísh to be used. And this is primarily through the act of translation. And my idea behind this is that the process of translating something into my language and then back into like English, for example, you have to make a certain series of conscientious decisions using the tools of íkjanísh that will intentionally that are meant to draw attention to some of the underlying stories or narratives that operate below the surface. So here I created a simplistic diagram. So you take a sentence like the village is planting trees. And in one way you can translate it into íkjanísh and to visibilize the problematic stories we live by, like hierarchism, objectification, restrictivism, or by making a subtle tweak in the grammar of the language through translation, you can present alternative articulations of that same sentence that foster better relations between and among human and other than human beings. To illustrate, so I take the sentence, the village is planting trees, and then to visibilize a potentially problematic story we live by, in one way it becomes this sentence, which is pronounced as follows. And so that means the village, which is considered an animate, like collective being, is investing in more trees, which are then inanimate. And that's relation is held in Oldanek there. And so it essentially an event of the village increasing the number of resources they have access to. Like that's their primary concern. It's just about human need, and the well-being and actual like impacts of the trees are not considered in this type of construction. And alternatively, we've got the sentence, and with that one, with Okalnek, the trees are considered animate, and that translates to like the animate village is helping more trees to grow. So it becomes more of a long-term type of relation and not just putting trees into the ground for later use. And the differences are in bold, so it's very simple change that produces this effect. And so the trees are animate for one, and then also there's an added future suffix here with íkjanísh, which signifies that this event is something that is meant to go on into the future. It's the start of something and not just a singular act. It's about a committed relationship between and among animate beings. Now, this is just kind of a simple example. I hope this shows some of the potential for íkjanísh as a constructed language and as a utility language, and how it can be used to change or draw attention to what certain sentences are saying, especially in the context of like environmental or climate policies. In my project, I specifically look at United Nations documents such as the Paris Agreement and the United Nations Environmental Program, and look at their language and see what kind of stories they're telling and ways it could be otherwise using íkjanísh as a tool. So thank you.
Thank you. Thank you. It was a wonderful talk, very interesting. So any questions, please? So while they are putting questions, what are you going to do after this? Yeah.
You have, for now, I'm gonna do some relaxing after this large process. But
yes.
Moving forward, one of the next things is to put together like a guidebook on my language. Because I want other people to be able to use it. And I originally was conceiving of this project as sort of doing that, but along the way, a lot of other things came up. And so I found that project like a guide, like exercises and instructions so that other people can learn how to use it and try it themselves. I also want to expand the vocabulary and continue with the grammatical structure, looking at other languages, both natural and constructed, and then applying íkjanísh to all sorts of documents and policies, and also in looking at especially looking at conversations that are kind of more positive too, and may have less of that problematic language that I critique in my project.
I have another question. How did you come up with this idea, íkjanísh?
Yeah, it was a long road for sure. It's definitely the culmination of my academic career. Like coming into Hampshire, I was studying like environmental studies and linguistics. I thought of them as separate, and then I found ecolinguistics kind of this intermediary. And I then thought of like, what would it mean to take ecolinguistics as such a literal point to create a language actually? Like, what would that mean? What kind of possibilities does that open up? Another question, or wait, hold on. This question.
You see, there's one up here. Yeah, is your language intentional, rooted, or not rooted in another language? Oh, that was one of the questions I was thinking. Etymology, asking, I think.
Yeah, so there's ways that it definitely follows from a lot of other languages. I looked at a lot of indigenous languages, seeing how I could learn from them and alongside them, especially in the conception of animacy or inanimacy. Specifically, I learned from like Anishinaabe languages, from like Robin Wall Kimmerer, who's written a lot on the idea of language and its role in human relations. But a lot of it to be seen, and not to be spoken fluently either. So it's always something that people come at in a new way, and have to really make these decisions and not just go along with their unstated assumptions, as we tend to do in our natural languages, or the ones that we fluently speak. Let's see.
That's wonderful. Lovely idea. Unique project. Congratulations. Yeah. Thank you very much. Yeah. So I had to go for the next speaker. I think there's one question. Any book recommendations? Yeah, that's it. Any book recommendation?
Book recommendation. Oh, that's a good question. Yes, actually, there's a key one that I cited in my presentation is Aaron Stibbe's. I'll put it in the chat. Let's see. Yeah, I'll put a couple in the chat. That's a great question.
Yes.
All right.
Thank you.
Thank
Yeah,
you.
the thing on the chat line, and I'll go to the next speaker. Thank you very much, and congratulations, Kai. The next speaker is Danny Britta. A very interesting title, Doggy Minds. So she's going to talk. Danny, how are you? Long time.
I'm good. How are you? It's been a long time.
Yeah, it's been a long time.
All right, so let me share my screen. Share.
Yep.
And present.
Make it bigger. Yeah.
There we go. All right, so this is my div. I called it Doggy Minds because the first class I ever took at Hampshire was called Animal Minds, taught by Mark Feinstein. And so I thought it was a nice little play. It was probably one of the more influential classes because I am always been in love with, animal behavior, all that fun stuff. And so it's Doggy Minds. I would say it's probably the closest thing I can describe it as is a giant lit review of like all the different sorts of training stuff that is out there. That I've compiled into one document, and there she is right here. Actually, I was super influenced to do this because, like a lot of people, I got a pandemic puppy. There she is right down there in the corner. Her name's Pancake. She's the love of my life. And so I wanted to figure out the best way to train her. And so I came at this in like at 3 angles. I wanted to look at the evolution of dogs, their natural biology, what might influence their behavior. And then I wanted to look at like how to improve a dog's welfare themselves, how to understand dogs, talk to them, whole 9 yards. So that way we can create a relationship between dog and owner. And then lastly, I wanted to look at different training methods and how they have evolved over the years. So let's jump right into it. History of dogs. All dogs descended from the gray wolf at least 15,000 years ago. Dogs were generally bred for specific tasks such as livestock, hunting, protection, etc. And while there are some like quote unquote ancient breeds, the migration and movement of people over the years has really made the gene pool a rather homogeneous and blended. And the concept of pure breed dogs didn't exist until a few hundred years ago. And now these groups are super separated and they're isolated gene pools, which is really interesting. Today, there's, depending on who you ask, there's over 400 different dog breeds. Some different groups have different numbers, but I'm going to go with over 400. And they all have different morphologies and behaviors. When you are artificially breeding animals, there's a 4-pronged approach. And if you select for one of the prongs, it's going to affect the other ones. And those prongs are behavior, face, size, and color. And we could see that with the fox domestication project that's going on. As they selected for foxes with lower flight distances, they got different coat colors and floppier ears, which I think is really cute and interesting. So that happens with dogs too. And there's a lot of debate on behavioral traits. Like everyone's heard that German Shepherds are more protective and Border Collies are more intelligent, and how accurate that is really unknown. The science is very lacking in uniformity, objectivity, and coverage of different breeds. And all the studies I've read, there's probably only been like 8 different breeds that have been looked at. And so we need to keep that in mind when we're talking about this. Oh, my thing's messing up. Hold on. Can everyone see?
It's a little out of whack, Donnie. It's a little to the left. Part of you. There we go. That's much better.
I
can't see you, but we can see your presentation now.
Okay. Well, wait. Oh,
there you are.
There we are. Yay. All right. So let's talk about dog, where they came from, the gray wolves. Alpha males, get rid of that concept. It is complete garbage. You could do a whole div on how the concept of alpha males has like generated this hypermasculinity that we see in our own culture today with sigma males and all that garbage. Toss it out the window. I don't want to think about it ever again. I don't want to mention it. Basically, these studies that happened in the 1930s, they took a bunch of wolves from a bunch of different parts of the country and shoved them all together. And in that setting, yes, they have very socially hierarchical societies, and they fight for alpha position, and then there's the alphas and the betas and yada yada yada. That doesn't happen out in the wild. In the wild, wolf packs consist of a breeding pair, so a mommy wolf, a daddy wolf, and their young offspring. And this is just affected culture more than anything possible. The first guy who came out with the whole alpha concept was like, please, I'm sorry, I didn't mean for this to happen. It's not what natural wolf behavior is like. And so we can't assume that natural dog behavior is the same way. We can't assume that dogs have this super urge to be hyper dominant or whatever. So let's toss that out the window, please. The next section is cohabitating with your dog. I could spend probably at least 4 hours talking about the language of dogs and themselves. Dogs have a universally understood body language. So that means no matter size, shape, color, they can all understand each other, which is cool and interesting. The most expressive parts of the dogs are their eyes, their ears, their lips, and their tail. And that's what they use. And there's 3 major groups of signals that they have. They have calming signals, which serve as a way to calm another party down or be a self-soothing behavior. There's also displacement signals, which is trying to get built up energy out of the body, and then stress signals, which is a dog undergoing stress. And this can calming signals can be one of the biggest points of contention in a relationship between an owner and a dog. I'm sure a lot of you have seen those videos of the dogs acting guilty and stuff like that, where they've like torn apart a bag and then they're like, "Oh, I'm so sorry." One of the biggest things that a dog does to calm another dog down is looking away, because eye contact between dogs is seen as like very aggressive. And eye contact for humans is a very big deal. It's seen as a sign of respect and everything. So let's say we have a situation in which an owner is yelling at their dog turns away, trying to calm the owner down. The owner takes that as a sign of disrespect and then is angrier at the dog and escalates the situation further. What this does is teaches the dog that their signals are being disregarded, and therefore they might act up further, which is why, like, when I see those super extreme versions of those videos of the dogs heavily displaying those calming signals, it's probably because in the past maybe that their signals have been disregarded and they're trying to amplify their signals. So understanding dog body language and understanding what your dog is trying to tell you can be a very huge help in just living with your dog. Another thing that dogs need is a lot of enrichment. There's 5 different groups of enrichment. Occupational, which is giving your dog a job to do. Some people do this with sports, like dog sports, frisbee, stuff like that. Physical is not exactly exercise. It's more about manipulating their physical environment. So that's usually done with toys. Sensory is about stimulating the 5 senses. Dogs' strongest sense is their snout, is their nose. So using scent work is a really big deal and can really help get those neurons firing. Nutritional is allowing your dog to express their natural foraging behaviors. Animals out in the wild spend a lot of their day foraging and just looking for food. And so if you give your dog just food in a bowl, that is negating like 70% of their day of what they're supposed to do. So a really helpful thing is you can hide food around the house. You can buy those puzzle feeders. You can use their kibble as training food, whole 9 yards. Just make them work for their meal, and that will help negate a lot of behaviors that problematic behaviors that can arise from boredom. And the last one is social. Dogs are social creatures. They love playing with humans. They love playing with other dogs. So I would say if you have a lot of problematic behaviors, such as barking, chewing, or something like that, check and see and offer one of the types of enrichment and see if that helps negate some of those problems. Also, these work for humans too. If you're feeling really crappy one day, think to yourself, have I satisfied one of the 5 groups of enrichment? And go out and give yourself a new experience and see if that helps. So the last part is behavioral training. So this is the meat of the project. So behavioral training, it started off with, Pavlov coining the classical conditioning, which the dog salivated with the bell because he rang the bell every time the dog was eating. And then B.F. Skinner coined the term operant conditioning, which basically is behavior controlled by its consequences or behavior manipulated by an outside force. Today, there's talk of 4 quadrants of operant conditioning. And before I get into this, I want you to separate any moral things that you think of when you think of positive or negative or punishment or reinforcement. That's not what it is. It's not a moral thing. So positive is adding something, negative is taking away something, reinforcement is encouraging behavior, while punishment is decreasing a certain behavior. So positive reinforcement, you're adding something to increase a behavior. That's what a lot of modern dog training is based off of nowadays, giving food in order for your dog to do a certain behavior. Positive punishment is what a lot of people think of when they think the word punishment. It's adding something unpleasant in order to decrease a behavior. Negative punishment is kind of like a timeout. I would say you, for example, you do really bad on a test, so your mom takes your phone away. So you, to decrease the behavior of you not studying, you lose your phone. And then negative reinforcement is taking away. I get confused in this myself every single time I try and discuss it. So from here on out, just for the purposes of this, punishment refers to positive punishment only, and reinforcement refers to the positive reinforcement that we always think of. So dominance-based training is totally focused on making your dog submissive based off of that alpha thing that we heard of earlier. Punishment does not work at all. Dogs that have been trained using reinforcement only versus dogs that are way more obedient and are able to recall tricks better than dogs who have ever been punished. Dogs that receive punishment have higher instances of separation anxiety, aggression, and fear-based behaviors. It also doesn't address the underlying behavior. Dogs, they can become habituated to punishment and require more and more harsh punishment overall. Veterinary Society of Animal Behavior advises owners to not use any sort of shock or pain-based related training. So why is it still used? Nowhere in the USA are trainers required to get any sort of certification. You can just walk up on the street and be like, I'm a dog trainer, give me your dog, I'll train your dog. And that's perfectly legal. So there's a lot of misinformation going out there. And a lot of people don't go to dog trainers at all. They get their information from books and TV and online. And when I say I've seen some horrible stuff on YouTube, I've seen some horrible stuff on YouTube under the guise of dog training. Also, a lot of the canine training is still done with primarily dominance-based training. There are constantly reports coming out that they're using techniques such as alpha rolls, where they roll the dog down and pin it down. It doesn't surprise me, as it is a mostly male-dominated place that they're using these types of training behaviors. There's been a lot of pushback that positive reinforcement is very womanly and very soft-hearted, and so a lot of these macho men won't do it on their canine dogs, which is whatever. And yeah, so there's no regulation on any of that. And it's really hard to get pertinent information. I mean, books came out in 2002 that said how to do it. So I'm running out of time. So I'm going to skip to how I think you should train your dog. Because behavioral issues are the #1 cause of death for dogs under the age of 3. And there are 2 main methods that I think are the best. One called Nothing in Life is Free, and the other one is called Plenty in Life is Free. They kind of go off of each other. Nothing in Life is Free is basically making your dog say please before they get anything that they want. So anytime they want to go outside, they have to sit, or anytime they want to get their food, they have to sit, et cetera, et cetera. It helps, you know, your dog look to you and realize that it has to do what you want before it gets anything. And Plenty in Life is Free uses a technique called SMART, which is see, mark, and reward training. You see good behavior, you mark the good behavior, you reward the good behavior, and your goal is to try and do that 50 times a day. And it's focused on providing leadership skills to owners and giving them a regimented training schedule that is scientifically proven to work. Yeah, that's my presentation. That was my div. Let me stop sharing. Oh, all right. Any questions? Thank you. I can grab my doggy. Come here. She doesn't want to move. Never mind.
Danny, so it was a great talk. And what are you going to do after?
That is a fantastic question.
Yeah.
I wish I kind of knew the answer to that. I mean, I know that a lot of people, especially with going back to work and having these pandemic dogs, they're not going to know what to do with their dogs after, you know, their dogs aren't going to be used to being alone all day. And so there's probably going to be a huge boom for people who can board and train dogs and stuff like that. And I might be looking to get into that, but unfortunately that like requires me to have a space in which I can do that. But the world's my oyster right now.
Okay. Thank you. That looks great. And I have a question. You know, I see that dogs are widely used in custom enforcement. You know? I can see in the airports, you know, and they are effectively used. What kind of training they undergo?
That is it's a good question. It completely depends on the organization that's training them. Again, a lot of those types of people are still using the force-based training and the dominance-based training because that's how they've been using it for 40, 50 years. You know, that's how they've been doing it. But since custom dogs are relatively new, I'm hoping that they're using positive reinforcement with that. But it would just be based off of doing the scent work, which is
the smell and the chemical smell, I guess.
Yeah, training scent work is super easy to do. So I don't think they would try and do that in a forceful way, but who knows?
Now, there are a couple of questions here. I am wondering about how you use this information to help in training your own dog.
Yeah, so a lot of the stuff, I mean, I trained my dogs always growing up, but I had wanted to look at more complex behaviors. The slide I skipped goes really in depth on how to train super complex behaviors and how to do that. So I was able to use that to get her baseline like training down. And then I'm working on like cool party tricks for her to do. And I want to get her into one of those dog sports eventually. And that does require some training. So I'll be going further with that. And one day I'd like to also make her a therapy dog, where she goes into places and just lets people pet her because she's adorable and she's very great with people. So yeah.
The last question, and I'll finish it. What experience with dog training did you also do for your Div 3? Cindy is asking that.
So yeah, I did an internship at a dog training place over in Granby, Elise's dog training place. And so I got to watch a lot of that done by a professional and see how it is set up and like how to, because a lot of dog training is how to train humans how to train the dogs. And so I was able to run my own EPEC this semester and train a few dogs that were on campus, which is really cool. Yeah, so I got that experience as well. Thank you, Cindy, for being on the meeting.
Thank you, Danny.
Thanks.
Yeah, thank you. Congratulations. Okay.
Thank you.
Yeah. Take care. Nice to see you all. Now I can see now the last speaker. for this session. Matt is waiting. Matt Hinderhofer is going to talk about the effects of cannabidiol, or CBD, on colorectal cancer cells. All yours, Matt.
Yeah, so for my div, I looked at the effects of cannabidiol or CBD on human colorectal cancer cells. And I wanted to start with a thanks to everybody, my committee, John and Elizabeth, and a thank you to Naya and Sarah and all of my family and friends, and especially all of the NS divs and also CBD for the grant and funding my project. So I've always been interested in studying cancer as a whole, it definitely due to the prevalence that it has in many of our lives. There was an estimated of 1,762,450 new cancer cases, as well as 606,880 deaths estimated in 2019 alone. And of those, about 100,000 of them were colorectal cancer, with about 50,000 new deaths from that. And just based off of this, I started with some starting questions of basically just what is cancer? How does cancer form? How does it spread? What do we know about cancer? What do we not know about cancer, and how can it be treated? And just when I started this research with colorectal cancer, I learned that there was little progress being made on it due to the difficulty of treatment and the heavy reliance on disease detection. And so to answer this question, I started by looking at some of the general hallmarks of cancer, which are some of the common traits seen in all cancer. And these are uncontrollable growth, resistance to cell death, evasion of growth suppression, formation of cancer vascular system, also called angiogenesis, the enabling of replicative immortality, spreading to local tissue and non-local tissue, so invasion and metastasis, avoiding of immune destruction, and deregulation of cellular metabolism. And they're thought to be caused by these two enabling hallmarks of inflammation and genetic instability. And so over here on the right, I just have a picture of how all of those, hallmarks and how they influence the tumor microenvironment. And then next to it are just different therapeutic options based off of these. So in my research, I focus mainly on sustained proliferation and resisting cell death, as well as the enabling hallmark of inflammation, which I'll get into a little bit. So uncontrollable cell growth or sustained proliferation is under normal cell conditions, the cell maintains homeostasis, which means that the specific tissue will keep a specific number of cells and maintain a proper function. But cancer is caused when there's, a lot of the time it's caused when there's a disturbance in this homeostasis. And it's commonly due to a mutation of a gene that is responsible for regulating cell growth and regulation. So it's commonly a tumor suppressor or oncogene. And next, I looked into the resistance to cell death. So when a cell is stressed, it undergoes programmed cell death. And cancer cells actually evade this mechanism and grow despite of the cellular damage. And this process is thought to occur due to several mutations, and it's a valuable target for cancer treatment. In this, I looked specifically on the intrinsic pathway, which you can see on the right, which is carried out in the cell's mitochondria. So for my research, I began with understanding the mechanisms of cancer, and then I used a lot of the resources available in the lab because of a lot of funding issues and a lot of other things. So I looked at colorectal cancer and looked specifically at HCT116 cells. And I looked at how can I kill these cancer cells specifically and not non-cancer cells, and attempt to learn something new from it. And just surrounding a lot of regulations, there is a lot of new regulations surrounding research on CBD after the Farm Bill of 2018. And I'm sure everybody else also sees how much media attention CBD gets as being a common cure-all for migraines, for pain, for anxiety, and cancer. So my question was, does CBD actually kill cancer cells? So I began with a bit of a literature review, and I found through all of this that CBD induces apoptosis in cancer cells due to dysfunction of the mitochondria, which is thought to be due to an increased production of reactive oxygen species, which may be due to an increase in calcium levels in the mitochondria. And I also commonly saw upregulation of PARP gamma, which is thought to be associated with inflammation and may suggest why CBD is commonly used for inflammation and pain management and things of the sort. And I also just found a very extensive list of upregulated and downregulated genes that I used for my research. So as mentioned, I found a lot of these genes that were responsible for the intrinsic pathway of apoptosis. So I looked specifically at how the mitochondria was being affected with CBD treatment. So to begin my research, I used a cell viability assay where I plated some cells in a 96-well plate and then used a stain a fluorescent stain. So, or not a stain, but just a fluorescent to see how these cells are being affected by CBD treatment, and if the cells are continuing to proliferate when I treat them with CBD in comparison to a no treatment control. And they were treated with either 50 micromolar CBD or a methanol control of an equal volume. And this was the data after 20 hours using 3 technical replicates and 5 biological replicants. So basically through this, we can see that the 50 micromolar treatment significantly reduced the viability of the cells compared to the 50 micromolar methanol treatment, as well as the increasing concentrations. I did 75 and 100 micromolar treatments too, and found that 50 micromolar treatment was the most significant, had the most significant effect, which was pretty interesting. And then this just kind of is a broken down graph so that you can see directly the effects of CBD or the methanol control. And then this is just a graph over time that I have from this data to show over time after 20 hours, the effects of the 50 micromolar treatment compared to the 50 micromolar methanol treatment, and shows that like after this 5-hour mark, we see that there's a pretty significant decrease in viability of the 50 micromolar treatment of CBD, but not methanol. And so I then looked at the, so then I took cells and I treated them with CBD, and I incubated them for 16 hours and then did an RNA extraction from them and then made some cDNA, and then I could look at the relative change in gene expression. And I looked specifically at cyclin D1, P53, BCL2, TRPV1, and I compared them to a tubulin loading control, and I did these all in triplicate. I looked at cyclin D1 because it's a cell cycle regulator, would tell me if there is any changes in cell cycle activity. And then P53 is a tumor suppressor, which is commonly associated with cancer progression. So also would tell me if anything was happening to the cancer cells. And then BCL2 is associated with the intrinsic pathway of apoptosis. And TRPV1 is an ion channel that is used for calcium regulation and is also associated with inflammation. So through this, I found a pretty significant decrease in cyclin D1 expression, and then also a decrease in P53, BCL2, and TRPV1 in comparison to this loading control, which shows that there seems to be a decrease in cell proliferation. So cells are dying when treated with CBD. And it's like through this, I can kind of make a similar story to the literature review that these cells are dying due to mitochondrial dysfunction and calcium regulation changes. And so after this, I wanted to look directly at how the mitochondria were being affected by the CBD treatment. So in A, there's a merge photo. In B is just the mitochondria. In C is the nucleus, the nuclei of the cell. And then D is the cytoskeleton. And this is all for a no treatment control. And we can see, and then, so this was compared to when I treated it with methanol. And same, this is the merge is A, the nucleus is B, C is the mitochondria, which is pretty dark, you can't see it too well. And then D is the cytoskeleton. And then I compared this to the 50 micromolar CBD treatment, which in this, we can see a pretty significant, there's some pretty significant damage to the cytoskeleton of the cells, which will definitely say that there is damage happening, as well as some mitochondrial dysfunction, which could suggest a production of reactive oxygen species or other just damage to the nuclei. But overall, this staining gives a pretty good example of how these cells are being affected by CBD treatment and shows that the cells are being killed by CBD. So I ran through that pretty quickly. I tried to leave some time for questions. Cool. Cool. Okay, so let me go through some of these questions. So I got asked, do you think CBD could be a good treatment for cancer? So CBD, I don't know if anybody's ever heard before that spicy foods and things like that are good for cancer. And CBD kind of works in a similar way, and it is actually more effective than capsaicin in killing cancer because of the decrease in inflammation. I don't know if it would be a great treatment because it doesn't, also, I haven't compared these to normal type cells, so I can't confirm that it happens in only cancer cells, but literature review says that it does. So I think that it is an effective treatment, but I would definitely need to do some more research on that. Stop sharing my screen.
Matt, are you going to present these things in a big forum, like conference or something like that?
I wasn't planning on presenting in a big forum. I had to do this for a lot of job interviews and stuff. I got to present this work, so that was pretty interesting. Yeah. And I would love to be able to do more research on this. I would love to do RNA research, to do RNA-seq to see the full list of genes that are being differentially expressed with this.
Yeah. Wonderful, because you got nice results up there. So congratulations. Yeah. So what are you going to do next?
So I actually got a job working at Harvard in a stem cell lab. So after this, I'm going to be taking a little bit of a break from doing cancer research and do some work with diabetes and producing insulin secreting beta cells. And then after that, I do hope to get back into cancer research and pursue a PhD specifically in cancer.
Yeah, there's a question, how would CBD get to cells in your body?
Yeah, so there is a lot of research that I looked at into which mechanisms CBD is used to get into your cells. And so it looked a lot into the CB receptors of CB1 and CB2 and also TRPV1. So this non-specific ion channel that I was talking about seems to be how CBD gets into cells. And it's actually a heat regulator. So that's kind of how I mentioned how it works similarly to capsaicin.
Is it a small molecule or large molecule? You know the molecular weight usually?
It's around, I think, 218. It's a pretty big molecule.
Okay. Kilodaltons.
Let me double check.
KDA?
It's 314 grams per mole. So it's a pretty big
Oh, okay. Oh, small. Relatively small molecule. Yeah. So is it easy to available this thing now?
CBD, it's still very expensive. I bought 1 milliliter of CBD and it was about $50. Some of them were around 150. So yeah.
Is that right? All right. Are there any questions to Matt? Thank you, Matt. If not, thank you, Matt. Congratulations. Yeah. Okay. And so this is the end of this session, and it was a wonderful session, and I enjoyed it, and I'm pretty sure all the audiences enjoyed that. And thanks for staying such a long session from 1 to 4:30, almost 4:30. You guys did a wonderful job, and our tradition was always we have a little toast. I don't know. We can do that next time. We can have a toast back again in person. But this is the best time in Hampshire. I'm listening to this Division 3 thing, talks, and I very much enjoyed reading and listening to your work. Yeah, so wonderful work. I know it's a hard work. Congratulations. And also thanks for all the panelists and attendees here for joining with us. And have a wonderful summer. If there's no other questions. Naya, you have anything to say? No? Yeah. So I think we can give a big clap. Thank you. Thank you, Karen. Thank you, Sarah. Thank you, Naya.
Pleasure.
Yeah, all the people who
Very
helped to put together this one. I know it's a big task, you know.
And thank you, Dula, as your one of your last official services.
Yeah, official the last one. Yes, I know.
We're gonna miss you.
This is my 34th Div 3 session. Yeah. You know, going into, you know, so that'll be fun. I enjoyed it. Yeah. Thank you. And I think we are good to go. Yeah. Okay. Thank you.